Question. So we move on to the next speaker, that is Dr. Singh. And the talk is on looking at the brighter side of vitiligo, decoding molecular mechanisms behind low skin cancer incidence.
Thank you, Professor Picardo. At the outset, I would like to thank the organizers for giving me this wonderful opportunity to present my work amongst this august audience. So today I'll be talking about a really, brighter side to vitiligo, because in the past 2 days we have been hearing about what are the negative connotations associated with vitiligo, and today we will see why, what is the molecular mechanism behind the lower skin cancer incidence in vitiligo. And why do I say this is very surprising is because when you look at the skin pigmentation levels, we see that there is an indirect inverse correlation between pigmentation levels and skin cancer risk. So basically, people with fairer skin tones are 70 times more likely to develop skin cancers compared to people who have higher melanin content. So with this knowledge, when it comes to vitiligo versus the healthy, the normally pigmented control population, we see that indeed the cancer incidence in these vitiligo patients is much, much lower compared to the normal ethnicity matched counterparts. And this has been shown in several epidemiological studies, as Dr. Vivek had also previously shown. So our study was to understand the molecular basis for this conundrum seen in vitiligo. So the study approach that we took was, we took the non-lesional biopsy and the lesional biopsy from 6 vitiligo patients who had stable vitiligo, and only took the epidermal part and did RNA sequencing. So this was a genome-wide RNA sequencing the approach we took, and we focused our attention to small RNA molecules called microRNAs. So these are RNA molecules which do not code for protein, yet they have the power to regulate a lot of proteins by sequence-specific binding. So the results showed that there were at least 17 microRNAs that were differentially regulated between the lesional and the non-lesional epidermis, out of which 12 were significantly down-regulated and 5 were significantly up-regulated. We chose one of the most down-regulated MIRs for our further studies, which was called microRNA 211-5P or MIR 211-5P. And what we found was using bioinformatic-based prediction that one of the possible targets of this microRNA could be sirtuin-1. So, and we also validated it using Western blot analysis that indeed the microRNA could regulate the protein expression of sirtuin-1. So now, what is sirtuin-1? So this is a special NAD-dependent deacetylase, and its main function is to control a lot of cellular processes, including aging and cancer. And it has been actually very interesting, previously shown to protect cells from UV-induced damage. So that was therefore an ideal fit for further studies. And our readout for the assays, since we were trying to understand the cancer-specific phenomena, so we measured 2 DNA damage assays. It's a gamma H2X is like a red flag. Whenever there is a damage in the DNA, there is a signal given to the cell that come and repair this damage. And the second thing we did was cyclobutane pyrimidine dimer. So the nucleotides get dimerized, and that also happens, and especially the CPDs are known for UV-induced DNA damage. So we measured both these, and what we found was the MIR 211, if you remember, it was down-regulated in vitiligo. But when we artificially overexpress it, we see a very high level of gamma H2X positivity. That means high level of DNA damage. But as soon as we co-treat it with the sirtuin activator, which is the resveratrol we have used in this study, and the DNA damage comes significantly down. The same results were also obtained when we understood, when we checked the CPD dimer formations. So the sirtuin-1 activator was able to bring down the DNA damage caused to the keratinocytes. So when we see the sirtuin-1 levels in vitiligo, to confirm whether this is actually true in the vitiligo scenario, indeed the lesional samples had higher levels of sirtuin-1, both at protein as well as mRNA levels. So the next question was, what could be the upstream regulator? So SIRT1 was there, upregulated, MIR211 was downregulated, but do we have, a master regulator for this? And for that, we studied the literature and found that long non-coding RNAs. So these are again RNA molecules which they can regulate microRNAs in a sequence-specific binding way. So they don't again code for proteins, but they have again a power to regulate a lot of proteins in the cells. So we found out that MALAT1, which stands for metastasis-associated lung adenocarcinoma transcript 1. So this was one particular long non-coding RNA, which was, we found that it could be important in regulation of microRNA 211 and thus SIRT1. So here what we did was, we either took MALAT1 wild type construct, which had the normal sequence for binding the microRNA 211, or we specifically mutated that sequence, and we found that the MALAT1 could bind MIR211 only in a sequence-specific manner. The moment you mutate the sequence, it was no longer able to bind to MIR211 and suppress its activity. So finally what we found was that whenever we artificially, when we overexpress the MALAT1 construct, the MIR211 was downregulated significantly, and this downregulation helped SIRT1 overexpression. So in a nutshell, if I have to explain you, in the normal cells, what happens is there is a basal level of MIR211, which is downregulating the SIRT1 by binding to the mRNA. But in lesional skin, what happens is the long non-coding MALAT1, this RNA is expressed so much that it is able to bind and suppress the MIR211. So now the MIR211 is no longer available to suppress SIRT1 mRNA, and therefore leading to high expression of this SIRT1 molecule. And this could be the main reason, one of the possible reasons why the keratinocytes, despite not having so much melanin, are still protected from UV-induced skin cancers. So this was published previous year in BJD, and I would like to acknowledge my students who have done this work, the research funding, and the various hospitals, the clinicians who are my collaborators, who have provided us the precious patient samples. Thank you for your attention.
Thank you very much for this very nice talk about
you,
the possible mechanism of controlling the UVB damage in vitiligo skin. It's very intriguing, this indication on the involvement of the SIR2 pathways, and that regulate also apoptosis of the cell and autophagy situation. So certainly it's involved in several recycling
So
mechanisms.
autophagy is another aspect we are looking at, and again that is also regulated. So it is another survival mechanism, and again the same kind of proteins are involved in that also, which is regulating this. But that is an ongoing study in the lab.
Question, comments, please. Professor Passeron.
Terry, why don't you take this one? No, I'll just take it after you.
Thank you.
No, just a comment and congratulations for your work. I think it's really important to provide such works because I think you are experiencing the same as me. We are giving talks to dermatologists and physicians who are not experts in vitiligo. And when we are talking about JAK inhibitors, interferon gamma blocking, UVB, and all of them, the first question they raise, they say, oh, it's, but what about the cancer, the skin cancer risk? And they say, but it's decreased as compared to atopic dermatitis or other indications. But there's still this thinking. So, okay, we explain them, for example, for the risk of melanoma, and they can understand. But what you just show is also explaining why we have also a decrease of other skin cancer. So I just wanted to thank you, and I think this kind of work are very important and needs to be explained to the community because many patients are not treated because of this fear. So thank you.
No, that's correct, because UVB is the mainstay of the therapy given to the patients, and of course they have this concern. But the good news, even better than this, is that even the chances of internal cancers is significantly reduced in these patients. So this is, you lose something, but you win something. Yeah.
So that is a very nice presentation. So first of all, I want to congratulate you on talking about microRNA. I don't think that was used as a research tool in any of the talks so far, so congratulations.
Thank you.
I was wondering, in your two populations, and coming from a multiracial country like the United States,
I really appreciate this talk because I have to deal with the exact same thing as theory is. And then on top of that, we're the only two photobiologists, I think, who do work in photomedicine. But there's this group of patients who are non-photoadapters in vitiligo who actually do have a phototoxicity to phototherapy. They're a minority, maybe 10, 15%, and it's not a large percentage, but it's there. And then there are a group of people within vitiligo who do get skin cancers. All of us have...
It is not all or none phenomena, of course. Right. But yeah, if you see a wide number of people, then the percentage is very significant. It's 3 times lower, the incidence of skin cancer.
So specifically, my question is, mechanistically, if you had access to those tissue, could you compare the levels of these microRNAs and citrulline? You could?
Yeah.
Okay. How many patients would you need to determine if this is the true variable that's causing the oncoprotective mechanism against skin cancer?
Okay, so when we did this study, this particular study, the protein analysis was done only on 10 patients, but after that, this has been like 1.5 years that the paper has been published. After that, we have done it on more number of samples, and still, I think 99% of the samples show higher expression of sirtuin-1. So that is a very good proportion. Yeah.
So again, thank you very much. If you no other question, it's a very interesting presentation. Thank you. So